Hello everyone, this is mini lecture 1.2, and during this mini lecture we're going to be talking about the history of microbiology. There are three learning outcomes for this mini lecture. First, you should be able to explain the relationship between technology and scientific discovery. Two, be able to explain the relationship between experimentation and scientific theory. And describe aseptic technique. So prior to the discovery of microbes, there were a number of ideas about how life was generated. And the popular belief at the time in the 1600s and earlier was this idea of spontaneous generation, that inorganic material could create life, and that there was this invisible life force that was present in matter and in the air that led to the creation of life. And even after the discovery of microbes, there was still some belief in abiogenesis by some scientists and the community. Biogenesis is the opposite of spontaneous generation. Biogenesis suggests that all living things can only arise from other living things of their same kind. So a mouse could only come from another mouse instead of dirty laundry combined with some grain and allowed to sit for a certain amount of time in the right conditions, producing a mouse, which is what spontaneous generation suggested. So there are a number of things that helped us to better understand where life comes from. And the first of those things was the invention of the microscope. Prior to the late 1600s, there was no technology that allowed us to observe cells or microorganisms. So just by strict observation with the naked eye, it's hard to explain how things like maggots suddenly appeared on rotting meat, or how mice suddenly appeared in the grain jar in your kitchen. Robert Hooke was the first to begin using microscopes, and he developed and improved the microscopes of his time. He studied a number of household objects and plants and trees, and he was the first to observe and name the structures that we call cells. And while looking at a piece of cork under his microscope, he noticed that there were small chambers or rooms that made up this structure, and they reminded him of the rooms in a monastery, which are called cells, so he named them cells, and we still call them cells today. Antony van Leeuwenhoek was also using microscopes at the same time as Robert Hooke. His microscope was very different than Hooke's microscope. Van Leeuwenhoek was not a scientist. He was a merchant in textiles, and he was used to using lenses to count threads in fabric. And he used his understanding of glass and lenses to develop a microscope that was very powerful. And he began looking at a number of things like water drops and scrapings on his teeth. He looked at his blood, he looked at semen, and he was the first individual to observe bacteria, and he called them animalcules because they reminded him of tiny animals. He observed protists in pond water, and he was the first to observe a biofilm by scraping the stuff off of his teeth and looking at it under a microscope. So because of the microscope, we had this understanding that there were objects organisms that were too small for us to see with our eyes. So there were some scientists that, with this understanding that microbes exist, attempted to disprove the common belief in spontaneous generation. And we're going to talk about each one of these scientists and their experiments and how they attempted to prove biogenesis and not abiogenesis. And the first scientist we're going to discuss is Francesco Redi. He was the earliest of these scientists, and he believed that life had to come from other similar life. So he designed this experiment to test where maggots come from. And he set up 3 jars. Each one contained a piece of meat. One jar was left open, one jar was covered with gauze, and one jar was sealed completely. Now, the belief in the past was that there was a invisible force carried in the air that could generate life. So the jar covered with gauze allowed the air to come in contact with the meat, but not the flies. The open jar allowed flies and air to come in contact with the meat, and then the sealed jar prevented anything from contacting the meat. And as Redi left these jars out, he discovered that only the open jar, where flies were able to come in contact with the meat, were able to produce maggots. And he hypothesized that the maggots had to come from the flies, that they were not generated from the air. However, Redi's experiment was conducted a little earlier than microscopic discoveries. So people weren't ready to believe that there were organisms too small to see that could contribute to life, like fly eggs. So a little bit later on, in the early 1700s, we have John Needham and Lazzaro Spallanzani. And John Needham conducted his experiment first, and he was a scientist who strongly believed in spontaneous generation. And his experiment is this one here. He boiled some chicken broth and corn mixture and left the flask on the counter. And his hypothesis was that Francesco Redi's experiment was incorrect, that given the right conditions, spontaneous generation could occur. And as he left the flask open, sitting on the counter and waited, sure enough, there was growth in his flask. And he concluded that spontaneous generation contributed to the growth in his flask. Well, Spallanzani saw this experiment, and he felt that there were a number of flaws in this experiment. So he decided to repeat the experiment, only he added this step. He used a second flask filled with the same broth, boiled them both. He left one flask open, just as Needham had done, but the second flask he closed. And he allowed the flasks to sit on the counter for the same amount of time, and he noticed that the open flask indeed have growth, but the sealed flask did not. But when he opened the flask, then there was growth in the flask. And he determined that there was something in the air, microbes could be carried in the air, and that those microbes were causing the growth in his flask. Well, those who opposed biogenesis suggested that the only reason this flask didn't have any growth in it was that he had killed the life force in boiling it. So we still didn't get a conclusion here with these two experiments. But Louis Pasteur, with his swan neck flask experiment, was finally able to put to rest this idea of biogenesis, and that organisms must come from like organisms, that life does not spontaneously appear from a combination of inorganic material. Louis Pasteur developed this flask, and it is called the swan neck flask because of the shape of the neck, and he boiled similar broth to Spallanzani in this flask. And in the first test, he left the neck intact, and the neck was designed strategically so that air could pass through the tube, but anything heavy like dust particles or pollen or microbes that were carried in the air would settle in the bend of the tube and could not be carried past that and into the broth. The second flask, he removed the neck and allowed it to sit for 3 days. And after 3 days, the first flask with the neck intact had no microbes, and the second flask did contain microbial growth. And Pasteur concluded and proved to the scientific community that there were microorganisms carried in the air that contributed to this growth. Pasteur's discovery helped to lead to some of the improvements that began to occur in medicine at this time in the late 1700s and early 1800s. A couple of doctors, Dr. Ignaz Semmelweis and Dr. Joseph Lister, both developed some practices that helped to reduce infection in medical procedures at that time. Ignaz Semmelweis was a gynecologist, and he discovered that if he washed his hands prior to birth, that the mothers were less prone to developing infection after birth. And he began to really promote the idea of hand washing and that practice to his colleagues. Joseph Lister took this even further and determined that if he disinfected his surgical area, that there was even a greater reduction in infection in his patients. So he developed this process of the aseptic technique where we reduce the number of microbes present in a particular area. And he used a phenol spray to disinfect the air in his surgical theater, and he disinfected surgical wounds after surgery, and he also was in the regular practice of sterilizing his surgical equipment. And both of these doctors discovered that there was a significant reduction in infection when they did these things. So that leads further into this idea of germ theory. And germ theory suggests that there is a direct connection between an organism and disease. And Pasteur, who invented pasteurization, made this connection between human disease and infection. And Robert Koch, who came a little bit after Pasteur, even took this farther and developed a series of steps that scientists and doctors could use to establish whether or not an organism was pathogenic, whether it was the cause of a disease, and what specific disease it caused. And Robert Koch used these steps to prove in 1875 that Bacillus anthracis was the causative agent of anthrax. So that is the end of our mini lecture. Make sure that you go through these questions and that you can answer them. And remember, you can